very curious to see how the online journal club works. So we're trying to introduce this concept of Histone Acetylome-wide association studies. And the first thing we tackled with that approach is autism, which is maybe a crazy thing to do for a few reasons. Okay, and I'll come to that in a minute, but it was kind of wildly ambitious when we started, but it worked out. All right, so the whole concept starts with GWAS, right? GWAS just came before anything else. And the idea is, what do you want to do? What do you do if you want to find out the genetic basis of, say, some disease, right? The causative genetic factors for some disease. You do a genome-wide association study. And so you take patient DNA and non-patient DNA, and you genotype them, and you look for some difference in allele frequencies or genotype frequencies or so on. And that's your genome-wide association study. And you say, okay, if there's a highly significant difference at some locus, then that locus is presumably causal for that disease or contributes to the risk for that disease. So that's GWAS, and highly successful. And then somewhat complementary, similar concept in a way, is EWAS. That's epigenome-wide association study. So there you want to say, what's the epigenetic basis of a human trait? Similar concept, but the biology is quite different. And one thing about epigenome-wide association studies is that essentially all of the epigenome-wide association studies in the past, and with one exception, were based on DNA methylation. So you're looking for DNA methylation differences between disease and control, right? So EWAS was, I mean, to some extent, epigenomics started with DNA methylation, and other aspects of epigenomics are more recent, right? So we wanted to move, look at other aspects of the epigenome. So our version of EWAS is a bit different. So, but first, how is an EWAS different from GWAS? It has some challenges compared to GWAS, but I would say those challenges are also opportunities, okay? So one challenge is that you need to profile the disease-relevant tissue. You don't care about the epigenome of, say, a blood cell if the disease you're looking at has nothing to do with blood, right? So, well, let's say autism, okay? It's not quite true that autism has nothing to do with blood, but autism is, mainly a brain disorder. So you could say, EWAS looking at blood for autism doesn't make sense. You want to look at brain, right? Ideally. So another difference between EWAS and GWAS is that GWAS is just about the genetic influence of disease primarily, okay? But the epigenome reflects both the DNA sequence and the environmental influence. So in a sense, it mediates both. And so it's a more complex and more higher level phenotype, molecular phenotype, right? It's not as simple. But so that's a challenge. It's also an opportunity because you also want to understand how the environment affects disease. And looking at DNA is not going to help you there. So looking at the epigenome could help you much more. And then the other issue is difference between GWAS and EWAS is that if you have a genetic association with disease, it's hard to argue that the disease caused the DNA mutation, right? I mean, if it's cancer, sure. But if it's not cancer, then it's more likely that the DNA mutation caused the disease, right? But if you look at the epigenomic changes between, say, disease and control, it's hard to disentangle. You can say it's because they had the disease that the epigenome changed. It's not like the epigenome caused the disease. So you want to, the interpretation is not immediately obvious for EWAS. And then, of course, like I said, it's been mostly DNA methylation rather than chromatin profiling. But there's no reason why you cannot extend the EWAS methodology to chromatin profiling, more broadly histone modification and so on. So that's what we did. We said, okay, let's extend EWAS to histone acetylation, and has not been done before as far as we know. So we said give it a name. It's a histone acetylome-wide association study by analogy to GWAS. So here you take the patient chromatin, you take the control chromatin, you do histone acetylation ChIP-seq, and look for differences in histone acetylation between disease and control. And histone acetylation is interesting. It points to gene regulation, so on. So you said this is complementary, you can say, to DNA methylation. So why histone acetylation, and specifically why H3K27 acetylation? Okay, because there are many histone acetylations you can look at. Well, so firstly, histone acetylation is interesting because if you have a GWAS variant, a regulatory, that's non-coding, so regulatory variant, it's likely to alter the function of an enhancer or a promoter, right? Maybe it alters a function of boundary elements. I left that out. But generally, it has to alter the function of a regulatory element, and that's how it can affect a phenotype, right? So histone acetylation marks both active enhancers and active promoters. And that's been known for a while now. So in a sense, it's secular mark of active regulatory elements. It marks almost all active regulatory elements, and I'll come to that at the end of my talk if I have time. It's also, K27 acetylation is also really nice because it correlates with transcription factor binding and it correlates with mRNA levels. So you can almost think of it as a linker between TF binding and mRNA. So it's more correlated with TF binding than mRNA is correlated with TF binding. It's more correlated with mRNA levels than TF binding is correlated with mRNA levels, and so on. So it's kind of an intermediary. So we decided to apply this, like I said, to autism. And so what is autism? I guess I don't need to say that for most of this audience, but it's a fairly common disorder. It's marked by repetitive behavior or behavioral rigidity and social and communication deficits as exemplified. So in terms of behavioral rigidity, so autistic kids mostly would have, say, repetitive behaviors like stacking or turning wheels or something like that, right? So affects 1% of the population approximately, perhaps even more. And the challenge of autism, and the reason there are no drugs for autism, none, right? And there are no GWAS hits for autism. Why is that? It's because autism has highly heterogeneous etiology, right? So it's estimated there are about 100 genetic loci causal for autism or contributing to autism, perhaps more. And it's not just genetics, there's also environmental toxins during gestation that are thought to cause autism, maternal infection during gestation that's thought to cause autism, and so on. So many causes. And because of that, it's even considered not even one disease or not even one disorder, right? Like people say it's a collection of many different disorders that have overlapping symptoms. So, and that's why, you know, if autism is 100 different things, how do you treat it as one thing? How do you come up with one drug for autism if it's 100 different things? And that's been the challenge. So is there anything in common? And clearly at the genetic level, there's not much in common, right? People have done GWAS on autism about, I think, 2000 versus 2000 and found no genome-wide significant hits in GWAS. So there's no genetic mutation that's a common genetic mutation that explains autism. So what can we do? So we said, okay, we were inspired by Dan Geschwin. So these are my collaborators, Dan Geschwin at UCLA and John Mill in Exeter. So we were inspired by Dan's paper in 2011, quite a while back now, showing that there are about 900 dysregulated genes, autism versus control, in the cerebral cortex. So in 2011, he showed that there is a common denominator, right? There are some things in common in autism. Although the genetics are diverse and the environmental factors are diverse, the gene regulatory consequences are somewhat coherent, right? That there's shared dysregulation in autism. So that's exciting. So somewhere between the DNA and the phenotype, at a molecular level, you're seeing coherence in autistic individuals. And if you're seeing coherence, then you can design drugs for it in principle, right? And you can design one treatment that'll help most autistic individuals. So that was what was exciting. So we said, okay, DNA gene expression is a bit you can say it's one level above epigenomics and it's closer to phenotype. Can we see something in common across autistic individuals at the epigenomic level, even lower, right? And that was the point of our study. So we did a Histone Acetylome-wide association study of autism spectrum disorder. This was our data set. Is a quite, it's a puny data set by the standards of GWAS, but by the standards of EWAS, it's quite all right. So we had in the prefrontal cortex, we had 40 versus 40. In the temporal cortex, we had 30 versus 36, and the cerebellum, 30 versus 30. So disease versus control. In all, and they were all greater than or equal to 10 years old. We did K27 acetylation ChIP-seq. And in all, we had 209 ChIP-seq data sets in the study. One thing that's key here is we didn't do, back when we were doing the ChIP-seq, it was common to do single-end 36 base pairs or perhaps single-end 50 base pair ChIP-seq because it was cheaper. But we went for paired-end 100 base pair ChIP-seq because we wanted to sequence the DNA under the ChIP-seq peaks. And I'll come to that in a while. Why did we want to sequence the DNA under the ChIP-seq peaks? But for now, let's say just ChIP-seq looking at chromatin acetylation, Histone acetylation. So what did we do with the data? We had a Histone acetylation ChIP-seq, 207 data sets. Started with QC, of course, and threw away the bad data sets and mapped the reads. And then we had two separate pipelines because there were two things we wanted to do with the data. One is, of course, called ChIP-seq peaks, and each ChIP-seq peak is presumably a regulatory element, either an active enhancer or an active promoter. And then take the union of ChIP-seq peaks across all 40 or I guess 60 or 80 individuals. And those are our regulatory elements. And then harmonize the data sets using GC normalization, quantile normalization, removing, regress out confounding factors like age and sex and technical confounding factors and so on. And then call differentially acetylated peaks. So this is kind of like finding differential gene regulation, okay? So which genes are differentially expressed between disease and control? That's a fairly common strategy. So by analogy, we said which ChIP-seq peaks are differentially acetylated between disease and control. And those peaks will presumably tell us what's wrong with individuals, what's wrong in their brains. And of course, these differentially acetylated peaks, some of them will be cause, some of them will be consequence. And a priori, we don't know which is cause, which is consequence of autism, right? But at least it narrows the search. Now,
sorry, I have a question. This is Andrea, Massachusetts General Hospital. So for like methylation-wide association study, there's been population-based study, and that has allowed to identify potential confounding factors. For example, smoking is a well-known confounding factor, and so every EWAS analysis will adjust for smoke as well. I wonder if there's been any population-based acetylation study, and if that can help to identify the right confounders to add in your model.
Yeah, that's a very good question. So I presume smoking has been determined to be a confounding factor in the blood epigenome. I am not aware of any studies that show that smoking is a confounding factor in the brain, but who knows? It's quite possible. So, but we did not have information on the smoking status for the individuals that we had. So we got the brain samples from the Harvard Brain Bank and I think one other brain bank. We didn't know whether they smoked or not. It is an interesting scientific question. So I don't think my slides has the list of confounding factors. We regressed out about 9 or 10 confounding factors.
Do you regress out age and death as well?
We regressed out age, we regressed out sex. What do you mean by death?
Well, these are post-mortem. So when are the autistic individuals tend to die younger than the others?
They do, but we threw away the very, so in fact, a lot of autistic individuals, they die before they're 10 years old.
Right.
Much more than non-autistic individuals. We discarded those. And for the remaining individuals, for the 10 and above, we regressed out age.
Okay. Okay, cool, thank you.
We looked at, I think, the agonal duration. It did not have any significant correlation with our data. We tested also post-mortem interval. It did not have any significant correlation with our data, but I think that's because we regressed out a lot of technical confounding factors, like number of ChIP-seq peaks, number of reads, some other technical confounding factors I don't remember. So I think the post-mortem interval affects the quality of the data, but we regressed out 3 or 4 data quality covariates. And after regressing those out, I guess there was no residual effect of post-mortem interval on our data. So in all, I think if you read the paper, it lists the covariates that we regressed out. There's a good number. But yeah, unfortunately, smoking, we don't know the answer. We don't have the data. So our second pipeline is what we call haQTLs, histone acetylation QTLs. So some of you might be familiar with expression QTLs. So expression QTLs are genomic loci, say SNPs or indels or something, that correlate with population variation and gene expression. And so the assumption is that the expression QTL is a genetic variant that affects gene expression in the population. By analogy, the haQTL is a genetic variant that affects histone acetylation, or at least correlates with histone acetylation in the population, histone acetylation variation. So that's why we did paired end 100 base pair sequencing. So we had a special statistical test that allowed us to ignore, to not need genotyping data. So we just took the paired end 100 base pair ChIP-seq reads, used them to call SNPs. So this is actually a pipeline that Rick developed. So he's at the Stanley Center now. So Rick developed this bioinformatic pipeline to call genetic variants. It's kind of like exome sequencing, okay? You can say that when you do ChIP-seq, essentially you're doing regulome sequencing, right? In exome sequencing, you're concentrating all of your sequencing firepower on the exons. When you do ChIP-seq, you're concentrating all your sequencing on the regulatory elements. So why not do it as paired and 100 base pairs, sequence the whole regulatory element, and then call variants in that. That's what Rick's pipeline does, and we use Rick's pipeline in this project. So first we call variants in the human brain regulatory elements, and then we use Rick's algorithm to say which of those genetic variants are correlated with histone acetylation. And it's a very powerful statistical test that he developed. Combines both peak height variation and allelic imbalance in one statistical test. So we were able to call histone QTLs in human brain, in those 3 brain regions, and also differentially acetylated peaks from the same data, even though we had no genotyping chip data and so on, just ChIP-seq data. So that's what we use the data for. And in terms of differential peaks, this is the kind of thing we wanted to find. Okay, so these are 2 gene loci, where in the upper locus, you can see that in the autistic individuals, there are some examples. You have a very nice ChIP-seq peak at the 3' end of the CHD2 gene, and virtually no ChIP-seq, virtually no histone acetylation in the control individuals. Right? This is a dramatic difference between autistic and control brain. And down below, you have the opposite difference in the AP1M1 gene locus. You have a very nice ChIP-seq peak, presumably an enhancer in control individuals, wiped out, completely wiped out in autistic individuals. So there's no enhancer there, right? So we found quite a few of these dramatic differences. Of course, they were not all so dramatic, but this is what we wanted to see, differences in the epigenome between autistic and controlled brain. And as I said, some of them are causal, some of them are the consequence of autism rather than causal. So then we said, okay, let's do this on large scale. How many ChIP-seq differences do we see, okay, between autism and control? We did this slightly differently from GWAS, and I'm going to address that now. Some people who are accustomed to GWAS are a bit uncomfortable with what we did next. Let me explain it. So we said, okay, autism is a highly heterogeneous phenotype, heterogeneous etiology. We don't expect all autistic individuals to have the same epigenomic aberrations. In fact, given the experience from genetics, people told us that we're wasting our time. There's no way we're going to find a common theme in even a subset of autistic individuals in terms of their epigenomics. So we said, okay, let's meet that lower bar. Let's see if we can find a subset of autistic individuals who have a common theme, who have a common epigenomic aberration. And so we said, we're going to discard the atypical autistic individuals, the autistic individuals who have atypical epigenomes. And what this is an XY plot. So we said for each epigenome, for each individual, we're going to say how far is there. So each dot is one epigenome, right? One individual, one brain sample. So x-axis is how far is that epigenome from control epigenomes. Y-axis is how far is that epigenome from autistic epigenome. Okay? So you can see that control individuals are quite far from other autistic individuals. Autistic individuals are quite far from other control individuals. And that's as it should be, right? But there are some things that cross over. So these red dots here are autistic individuals whose epigenome is quite close to control. And these blue dots here are control individuals that are quite far from autism. Okay, so they are the epigenomes that just have the wrong behavior for whatever reason. But what's really dramatic is that the blue dots are mostly here. The control individuals are mostly similar to each other. Red dots are mostly here. The autistic individuals are mostly like each other. And that's quite dramatic, right? So what we're seeing is at the epigenomic level, we're seeing a signature of autism. And this is nothing, right? This is like 40 individuals versus 40 individuals. Whereas in GWAS with 2000 versus 2000, you still see nothing statistically significant. You still see no signal. So this is quite dramatic. What these figures are saying is that, yeah, sure, there are atypical individuals who don't behave like their cohort. But on the whole, autistic epigenomes look like each other. Control epigenomes look like each other. Even when you have a puny 30 versus 30 or 40 versus 40 cohort. And that is a huge difference. So despite the fact that the etiology, the genetic mutations that cause autism are diverse, the environmental factors that cause autism are diverse, by the time they influence the epigenome, they're all influencing the epigenome in more or less the same way. So that's quite dramatic, right? You know that the genetic etiology is diverse, the phenotype is coherent, it's the same phenotype. At what point in that pyramid from the DNA to the phenotype, that convergent, do they all start looking like each other? So our data suggests that straight away, when you go from the DNA to the epigenome, they already start looking like each other. And that is very exciting. So you're seeing at the molecular level, epigenomic level, some coherence in autistic individuals. And then you can imagine that, okay, so let's start perhaps designing drugs to reverse that coherent aberration. That's the message. But to define differential peaks, right? Just like you define differential genes between autism and disease and control, we define differential ChIP-seq peaks between disease and control, but we threw away these, what we call outliers. So we threw away the blue dots that lie over here. we throw away the red dots that lie on this side. And we said these blue guys here are the typical controls. These red samples here are the typical autism. And we're going to only look at differences between typical autism and typical control. So it changes the question slightly, but it gives you a, you're looking at the coherent subset, and it gives you a larger number of differential peaks between autism and control. So in all, we got, a good over 5,000 differential peaks in prefrontal cortex and temporal cortex between autism and control. In cerebellum, we got only 250 differential peaks. This is not too surprising. My colleague Dan Geschwin has done transcriptomics on these same 3 brain regions, and he actually found 0 differential gene expression in cerebellum between autism and control. And he found about 900 differential genes between prefrontal cortex and temporal cortex between autism and control. So it's qualitatively concordant. So what this is saying is that the dysregulation that's happening in autism is only in a minor way in the cerebellum. Mostly it's in the cerebellar cortex. And this is a principal components analysis plot. Here again, you can see that the first principal component of our ChIP-seq data to a large extent explains the difference between autism and control. So blue is control, red is autism. Right? So then we said, okay, let's take these differential peaks in the cerebral cortex and try to understand their properties and what is that telling us about autism. So firstly, we said, does differential acetylation correlate with differential expression? Because Dan's lab had done differential expression, and so they have a paper that just came out in Nature, I think in December. And that's a nice paper to read alongside this paper. So unfortunately, differential acetylation and differential expression don't correlate that well. As you can see, the correlation coefficients are in the region of 0.3, okay, 0.33, 0.38. Above 0, they're positive, wonderful. And the correlation is statistically significant. So we use Fisher's exact to say, if the acetylation is up, how likely is it that the expression is up? And that correlation is statistically significant but weak. Why is that? Partly biology, partly measurement artifacts. So you can imagine in biology and molecular biology, right? You measure the same thing with 2 very, very different assays, ChIP-seq and RNA-seq. You're not going to get concordant results, especially when you're looking at differences, right? When you're looking at the delta between autism and control, it's already noisy. So you're taking one noisy signal, correlating it with another noisy signal. Some loci will be detectable based on ChIP-seq. Some loci will be detectable based on RNA-seq. Only a subset of loci will be detectable based on both. But so that explains the relatively weak correlation, but it's still statistically significant and in the same direction. The other way we looked for concordance, and we said, okay, we take our differential ChIP-seq peaks in temporal cortex and differential ChIP-seq peaks in prefrontal cortex. And you say, do they overlap? And they certainly have a lot of overlap, right? So that's a very nice validation that these are 2 independent brain regions. We're detecting more or less the same loci as differential in ChIP-seq in those 2 brain regions. So again, that's confidence building that, this is non-random. And then we said, okay, now we believe our ChIP-seq, differential ChIP-seq peaks between autism and control. What are they telling us about the biology, right? What's different between autism and control in terms of gene ontology and so on? And so what this is saying, if you look at the upregulated genes, or PFC up and TC up, are the loci that have higher acetylation in prefrontal cortex and temporal cortex of autistic individuals. And these are not too surprising. If you look at PFC up, you see, ion channels, you see synaptic genes, you see epilepsy genes, and so on. Well, yeah, not too surprising. Those are known to be related to autism. Temporal cortex, again, you see epilepsy and synaptic genes and so on over here. You also see a couple of novel, interesting, glutamate pathway, also not surprising for autism. Zinc ion homeostasis, that's kind of interesting, but there are some mouse models of zinc homeostasis genes that are appear to have something to do with autism. But if you look at the down-regulated loci, right, the loci that have lower acetylation in autism, those are pretty interesting. They, you see something new, okay? You see chemokine pathway loci that have lower acetylation in autism. Not very well studied in autism, but CX3CR1 is, if you knock that out, the mice have autistic symptoms that overlap with autism. So that's interesting. Histone deacetylase activity, I'll come to that in a minute. HDAC4 is a syndromic autism locus, okay? So it's known that HDACs have something to do with autism, but HDAC4 is not the only locus we found that has lower acetylation in autism. So this is telling us something about the major themes of what's changed in the autistic brain. So then we said, okay, how does this, we've looked at the epigenetics. How does it connect to the epigenetics of autism? Okay, so sorry, how does this, we looked at the epigenetics, how does this correlate to the genetics of autism? Specifically, right, some of our individuals, our brain samples, had Dup15q syndrome. So they had a duplication in the Q arm of chromosome 15. And that is a well-known syndromic form of autism. So they have some dysmorphology and they also have autistic symptoms. But, you know, there are a lot of syndromic forms of autism. They have autism and other symptoms. A priori, we would, I mean, I always felt personally that it's a bit weird to call that autism, right? Because they have so many symptoms and there's one gene that's knocked out and is doing so many things. Is that really even autism? At the molecular level, are they similar to idiopathic autism? And by idiopathic autism, I mean autism for which you don't know the cause. Now, x-axis is a ChIP-seq peak height difference, fold change in these syndromic autism individuals. Y-axis is the ChIP-seq peak height difference in the idiopathic autism individuals. And you can see that they're highly correlated, okay? Correlation coefficient of almost 0.9. Now that is dramatic. So what is this saying is that in the whole genome across thousands of loci, the ChIP-seq, the chromatin state has changed in the same way in these syndromic autism individuals as in the idiopathic autism individuals. What it's saying is that these two seemingly very different flavors of autism are really the same. And actually, if you look at the slope of this scatter plot, you can see that the slope is less than 1. That means the syndromic autistic individuals have more ChIP-seq fold change than the idiopathic autistic individuals.
Shyam, that's Dennis.
Yeah.
One question, maybe might be very stupid, but can this be attributed to the duplication and association with autism, or could it be like any CNV which basically doesn't do a thing, which would give a similar signature?
So the blue dots, look at the blue dots. The blue dots are not in the duplicated locus.
Ah, okay. Oh, okay. Thank you.
Only the red dots are in the duplicated locus. So even the ChIP-seq peaks that are not in the duplicated locus have more aberration in Dup15q than in idiopathic. So genome-wide, not just in the duplicated locus, genome-wide, the ChIP-seq change is greater in the idiopathic individuals, in the syndromic individuals than idiopathic. And that, but correlating in the same direction. So what this is saying is that at the epigenomic level, this Dup15q individuals, they're the same as idiopathic, only more in that direction, you know, slightly worse. And here's another example of convergence between syndromic and idiopathic autism. So what these rectangles here show are ChIP-seq peaks that are downregulated in autism, mostly in idiopathic autism, okay? And they're highly concentrated in this HDAC4 locus, which is mutated at a DNA level in syndromic autism. So this is so what this is saying is that in idiopathic autism, HDAC4 is mutated at the DNA level. In syndromic autism, HDAC4 is downregulated at the chromatin level. And so presumably that chromatin downregulation is causal for autism, right? And this is a nice way you can separate cause from consequence, where essentially you can say at some level, all autistic individuals have loss of HDAC4. The syndromic individuals have complete loss of HDAC4. The idiopathic individuals have partial loss of HDAC4 due to epigenetic changes. And so this is a very nice, so basically all these syndromic autism loci, which seemingly tell you something only about these very rare subsets of autism, actually they tell you something about idiopathic autism also. They tell you about all autistic individuals, right? And so again, we're talking about what's common, and epigenomics is telling us what's common. So part 2 is we said, okay, can we get to the genetics? Can we find some SNPs that affect ChIP-seq peak height? And if so, you assume that they affect gene regulation. And perhaps that can explain some of your GWAS signals. And here we use the bioinformatics developed by Rick to find these statistical associations. And what we're looking for is loci like this, okay? So here's a SNP, it's a GC SNP. The GG homozygote has a very tall ChIP-seq peak. The CC homozygote has a very small ChIP-seq peak, so presumably that mutation reduces the activity of that regulatory element. And the GC heterozygote has an intermediate height ChIP-seq peak, but most of the ChIP-seq reads come from the G allele, not from the C allele, because the C allele is, reduces activity. So it makes sense that the C allele contributes less to the ChIP-seq peak. So you have fewer C reads that overlap the C allele than overlap the G allele. So Rick developed this G-side test, which detects both this overall correlation between ChIP-seq peak height and genotype, and the allelic imbalance at heterozygotes, and combines those two to get more statistical power. So based on the G-side test, we detected about 2,000 histone acetylation QTLs, SNPs that look like this, in each brain region. And note that these are not autism QTLs, these are brain QTLs, okay, because we regressed out the effect of autism. So in principle, they tell us how the SNPs affect brain function, and they could be causal for any brain-related phenotype. So here's an example of GWAS SNP that has a joint effect on 5 psychiatric disorders, genome-wide significant. And it's in LD with this SNP, RS476, etc., that, as you can see very clearly here, is associated with ChIP-seq peak height. It's also associated with allelic imbalance at heterozygotes. So presumably this SNP is a regulatory SNP that affects the function of this ChIP-seq peak. And it's in strong LD with a GWAS SNP for psychiatric disorders. So presumably this is the causal SNP for these 5 psychiatric disorders. Here's another example. Here's a schizophrenia GWAS SNP in the GRIN2A locus, which is a fairly famous locus. Again, a very strong ChIP-seq QTL or histone acetylation QTL. You can see here from the box plot of the peak heights for the various genotypes. And this is a SNP that is an LD with the schizophrenia GWAS SNP, and presumably the causal SNP. So that's the beauty of this approach. You get differential ChIP-seq peaks and you get regulatory SNPs. So to summarize, right, this is our model. Autism spectrum disorder is highly diverse in terms of etiology.
Sorry? Sorry, it took me a while to unmute. Could I interrupt, please, and go back one slide, please? So how, I didn't quite understand how you found that this was the causal SNP, if there could be another SNP in LD that might also be causal?
Right. So let me clarify. This is not a causal SNP. This is a candidate causal SNP.
Okay.
So for each GWAS SNP, you'll have lots of SNPs that are in LD.
Yeah. Okay.
One of them is causal.
Okay. Okay. I thought maybe that you had a special method to
no.
prioritize that.
Okay. So this is a method to prioritize, but it's not a method that proves that it's a causal SNP. For that, you actually functional assays. But
that's why I was wondering.
Exactly. So it's a normal thing when you have a GWAS SNP, you find an EQTL that's in LD with your GWAS SNP, and then you say that's probably the causal SNP, but you want to validate that.
Mm-hmm. Yeah.
Same thing with acetylation QTLs.
Okay.
Now the beauty of histone acetylation QTLs is that you have more statistical power, and Rick showed that in his Nature Methods paper a couple of years ago. That you can take a cohort of, say, 50 individuals and do expression QTL analysis. You'll get, say, 800 or 900 expression QTLs. You can do histone acetylation QTL analysis. You'll get 8,000 or 9,000 histone acetylation QTLs. And that's the beauty of this statistical approach. Because in expression QTL analysis, you cannot look at allelic imbalance so easily. With this method, you can look at allelic imbalance that gives you most of your statistical power.
I have a question. If you take the different peaks between autism cases and controls, those that are significant, and you look at the overlap between these peaks and GWAS region candidated from, for example, schizophrenia, do you see an enrichment or not?
Not for schizophrenia, but because our study was autism. So
yeah,
I assume you're talking about the entire list of 5,000 differential peaks, right?
Right. Yeah, the significant differential peaks. Are they overlapping any, or are they close to genes that, for example, are candidates to schizophrenia or neurodevelopmental disorder in general?
Okay, so our study was on autism, so presumably they should be near genes that are autism genes.
Right, yeah, sure.
So if I remember correctly, we found a statistically significant enrichment for being near Safari autism genes.
Okay.
Or for being near exome sequencing autism genes. I don't remember which of the two. We looked at 2 lists of autism genes and we found a statistical enrichment near one of those lists.
Okay. I think this is important because that will highlight that the global change that you see between autism and control is somehow related to autism rather than some other environmental factor which might be just more prevalent in autism individuals.
Exactly, yeah. So we did find an enrichment near one of our autism gene lists. I forget which one. So, and of course, we found an enrichment near the gene categories that are well known in autism, which is synaptic transmission, immune, ion channel, and so on, and epilepsy. So, and also we found an enrichment near genes that are upregulated at 12 months postnatal, at 1 year after birth. And again, those are synaptic genes, so they're upregulated at that time point. And it makes sense for autism. So here's our model. Autism has diverse etiology. It has, coding exome changes. It has copy number variants that cause autism, the environmental toxins, maternal infections, so on. So very diverse. But they all converge on the same phenotype, okay, or overlapping phenotypes, which is those 3 phenotypes those 3 diagnostic criteria I mentioned. At what point do they converge? Do they converge at the level of the tissue? Do they converge at the level of the cell? Do they converge at the level of protein, RNA, epigenome and so on, as you're getting closer and closer to DNA, right? So our results suggest that they converge very, very fast. They converge at the level of chromatin changes. So this is exciting. So at the level of chromatin, you're finding common denominator in autism, and maybe these chromatin changes, at least some of them, they mediate the effect of, they mediate the causal relationship between all of these things and autism. So you don't need a drug to target any specific coding change. Instead, if this chromatin changes are mediating all of this, right, and the chromatin changes are common, you can come up with an autism drug that tries to reverse the chromatin changes instead of trying to reverse this. And that's really important because no drug company is going to go after autism CNVs or autism coding changes. They're too rare. There's no money to be made. But, you know, that's a reality. But if the chromatin changes are common, the drug company is interested, right? Because you can come up with a drug that can help a large fraction of autistic individuals. And that, I think, is the concrete implication of our work. So to summarize, this is the first cohort scale histone acetylome-wide association study. We found thousands of common epi mutations in autism, even though there's 0 common genetic mutations in autism, at least what's known. And then also the epi mutations were more or less the same in idiopathic autism and syndromic autism, which is quite dramatic. So that's another level of unification across the autism diversity. And then the dysregulated loci, some of them were the usual suspects: ion channels, synaptic, epilepsy, glutamate receptor, and so on. Some of them were a bit more interesting: HDAC activity, chemokines, morphogenesis, which all have some vague role from mouse studies, but they're not so well known in autism. Okay. And then finally, we found a whole bunch of haQTLs, histone acetylation QTLs in each brain region. And 4 of them are in LD with known psych GWAS loci. And of course, psych GWAS is expanding and expanding and expanding. So there are going to be GWAS studies for psychiatric disorders that are 10 times larger than what exists today. And so the number of GWAS SNPs for schizophrenia and bipolar and major depression, autism is going to just explode. And when that happens, these 2,000 haQTLs will be very, very useful to prioritize causal candidates for those GWAS loci, because most of them, again, we expect will be non-coding. So, and that's it. We're out of time, so I'm going to skip the second part of my study and jump straight to the acknowledgements. So, most prominently, I'd like to acknowledge Wenjie, who did the bioinformatics for this study, and Jeremy, who did all of the experimental work for this autism study. And they were assisted by Shreen and Lisha on the wet lab side. And Rick, of course, developed a lot of the bioinformatic methods for this. And Vibor also developed some of the other ChIP-seq calling, peak calling bioinformatic methods. Also, like to acknowledge my collaborators, Dan Geschwin and Neil in his lab, and John Mill from Exeter, and Chloe Wong from King's College. And for the haQTL approach that Rick and Jeremy developed, so Martin and CC, those are my colleagues at GIS. Yeah, that's it. Thank you.
Thank you very much. If you want to, you can stop sharing the screen. Are there any questions from the audience?
Dennis, I have a question.
Sure, go ahead.
So this is Jacob, I'm a PhD student at MGH. What is the purpose or logic behind discarding those samples of the patient who died at 10 years or younger? Wouldn't they contribute to a strong phenotype?
Yeah, that's a good question. The thing is, the brain epigenome, the brain changes very dramatically after 10 years. That's